Neuroscience enthusiasts today we're diving into fascinating world of how our cells manage their DNA, regulate gene expression, and even how tiny modification can lead to big biological effects. First up, let's talk about DNA. Think of your genetic material as a super long, tightly packed spaghetti noodle, except in our cells, the spaghetti is organized into a structure called chromatin. Chromatin is made up of DNA wrapped around histone proteins, which act like spools. These histones aren't just passive spools, they get decorated with all sorts of chemical marks. Modifications like acetylation, methylation, phosphorylation, ubiquitination, even lysine benzylation. These chemical tags are like tiny post-it notes that tell the cells whether a gene should be active or silenced. These modifications are the cell's way of saying, hey, this gene is important today. For example, acetylation of histones often leads to a more relaxed chromatin structure, making it easier for the cell's machinery to read the genes. Think of it as opening the curtains for a big performance. Conversely, methylation can tighten things up, leading to heterochromatin formation and gene silencing. What happens when things go wrong? Well, sometimes DNA suffers a double strand break. The spaghetti noodle is snapped in half. If these breaks aren't repaired properly, they can lead to genomic instability, which is a fancy way of saying cellular chaos. To prevent this chaos, cells can activate apoptosis, or programmed cell death. It's like the cell's way of saying, I've had enough, a crucial process to prevent damaged DNA from causing trouble. In histone modifications, the histone deacetylases, sometimes called HDACs, are like the cell's version of a bouncer, removing acetyl groups and tightening their chromatin. Interestingly, scientists have developed histone deacetylase inhibitors, the cell's relaxation pills that can reactivate silence genes with potential applications in cancer therapy and epigenetic research. Epigenetics is all about these chemical marks, like the methylation patterns and histone modifications that influence gene expression without changing the underlying DNA sequence. It's like decorating your house differently without changing the structure. Sometimes these modifications are influenced by microbiome metabolites like short-chain fatty acids, which are produced by our gut microbiota. These molecules, such as sodium butyriate or phenylethyl alcohol, can modify histones and impact gene expression in our gut epithelial cells, affecting phenotypic plasticity. Bioactive phytochemicals like curcumin or anacardic acid found in turmeric and other plants can impact histone modification and gene transcription. They're like nature's little molecular tools influencing epigenetic marks and potentially offering health benefits. Let's not forget about telomeres, protective caps at the end of our chromosomes. Disrupting telomeric silencing or shortening them can lead to cellular aging and genomic instability. In the end, understanding these tiny chemicals, tags, and cellular processes not only reveal the elegance of biology, but also opens doors to innovative therapies and health strategies. Because sometimes a small change can make the biggest difference, whether it's tweaking, methylation, or phosphorylation event, or a simple histone modification. That's the beauty of the microscopic universe in science. Complex, dynamic, and endlessly fascinating.